Humanity has dreamed of living forever for centuries. Think of ancient myths, fountain of youth, elixir of life. These stories span cultures and continents, all sharing a common, deeply human desire to overcome the limits of our own biology. For most of history, this dream was fantasy and legend. We saw aging as an inevitable force of nature, a one-way street leading to decline and eventual death. What was once a subject for poets and philosophers has now become a question for scientists and researchers. We are living in a revolutionary era where the tools of modern biology are finally allowing us to understand the fundamental mechanisms of aging itself. Could biological immortality, or at least a radical extension of human health span, be achievable within our lifetimes? Specifically, we will investigate the scientific basis for the prediction that we might see these breakthroughs in the 2040s or 2050s. This is not science fiction. This is based on real, tangible progress happening in laboratories around the world right now. The journey from myth to a solvable scientific problem has been a long one, but we are now standing at a critical juncture where the dream of extended life is beginning to look like a potential reality, built on a solid foundation of biological discovery. To understand this incredible possibility, we will follow a clear path. We will start by briefly looking at the history of this dream and how science began to tackle the problem of aging. Then, we will examine the key discoveries of the 20th century that laid the groundwork, such as our understanding of DNA and cellular limits. From there, we will dive into the current cutting-edge breakthroughs in life extension research, leading to a detailed look at one of the most promising technologies, cellular reprogramming, using what are known as Yamanaka factors. This remarkable process has shown the ability to reverse signs of aging in cells and even in animals, offering a potential pathway to rejuvenate human tissues. Finally, we will look ahead, laying out a possible timeline of milestones leading up to the 2050s. We will consider what it would take to get from today's laboratory experiments to widely available therapies that could halt or even reverse aging. Of course, such a profound change would come with significant risks and ethical questions, which we will also address. The goal is to provide a balanced, informative, and realistic assessment of where we stand on the threshold of potentially rewriting the human story. The question is no longer if we can intervene in the aging process, but how and when. The next few decades could provide the answer. The 20th century began with a monumental question: what carries the instructions for life? This all changed in 1953. James Watson, Francis Crick, Rosalind Franklin, and Maurice Wilkins unveiled the structure of the deoxyribonucleic acid molecule, or DNA. Their discovery of the double helix was the key that unlocked the very language of life. It showed how genetic information could be stored in a simple code, and crucially, how it could be copied accurately from one generation of cells to the next. This groundbreaking discovery immediately paved the way for understanding the central dogma of molecular biology: DNA, RNA, proteins. Proteins are the workhorses of the cell, carrying out the vast majority of its functions. Understanding this process was like learning to read the instruction manual. It meant that scientists could finally begin to connect specific genes to specific functions, and more importantly, to specific malfunctions that occur during the aging process. In the early 1960s, a biologist named Leonard Haylick made a startling and crucial observation. He found that normal human cells would divide a finite number of times, roughly 50 times, before they entered a state known as senescence. This phenomenon became known as the Haylick Limit. The answer began to emerge in the 1970s. They discovered that the ends of our chromosomes are capped by protective structures called telomeres. The researchers found that with each cell division, a small piece of the telomere is lost. Eventually, the telomeres become critically short, signaling the cell to stop dividing. However, the story had another twist. The same researchers also discovered an enzyme called telomerase. This remarkable enzyme has the ability to add length back onto the telomeres, effectively rebuilding them and resetting the cellular clock. In most of our body's cells, telomerase is turned off, but in stem cells and cancer cells, it remains active. This discovery was a double-edged sword. It revealed a potential path to cellular rejuvenation, but also highlighted a deep connection to cancer. One of the most significant discoveries in this area came from a surprisingly simple intervention: caloric restriction. The rats on the calorie-restricted diet lived dramatically longer, up to 40% longer. This finding was revolutionary. It could be slowed down. These experiments were repeated over the decades in a wide variety of species: yeast, worms, flies, mice, and even monkeys. This demonstrated that aging and age-related diseases were deeply intertwined and that both could be influenced by metabolic pathways sensitive to nutrient intake. The hypothesis was that caloric restriction works by shifting the body's resources away from growth and reproduction and toward maintenance and repair. By the end of the 20th century, aging was understood as a complex but malleable biological process. The stage was now set for the 21st century, where researchers would begin to develop drugs and therapies aimed at directly manipulating these pathways to extend human health. Scientists in the 21st century began to focus on what happens after cells stop dividing. These senescent cells accumulate in our tissues as we age. They actively secrete a cocktail of inflammatory proteins that can damage surrounding healthy cells. This realization led to a powerful question: what if we could selectively remove these troublesome cells? That idea gave rise to a class of drugs called senolytics, designed to hunt down and eliminate senescent cells from the body. The initial results from animal studies were nothing short of spectacular. Those mice lived longer. They stayed healthier, more active and robust even at advanced age. Human trials for both senolytics and autophagy boosters are now underway. The ultimate goal is preventing a wide spectrum of age-related decline, but the near-term focus is safety and efficacy in targeted conditions. Could we achieve the same benefits without the extreme diet? This quest led to the development of caloric restriction mimetics, drugs that trick the body into thinking it is in a state of fasting. Two of the most studied drugs in this category are rapamycin and metformin. Metformin, in particular, is the subject of a landmark human clinical trial called TAME. This trial aims to see if metformin can delay the onset of a whole cluster of age-related diseases in healthy older adults. Another critical piece of the metabolic puzzle is a molecule called NAD+. As we age, the levels of NAD+ in our bodies decline precipitously, which is thought to impair sirtuin activity and contribute to the aging process. This has led to a surge of interest in strategies to boost NAD+ levels. Researchers are actively studying various precursor molecules. In animal studies, supplementing with these NAD+ boosters has shown remarkable effects, improving mitochondrial function, enhancing muscle stamina, and in some cases, extending lifespan. The development of the CRISPR gene editing tool has been a complete game changer. It provides a relatively simple, cheap, and precise way to make specific changes to DNA. Alongside gene editing, advances in stem cell therapy represent another powerful front in the fight against aging. Stem cell therapies aim to counteract this by introducing new, healthy stem cells into the body to replenish the depleted reserves. The combination of these two technologies, gene editing and stem cells, is particularly powerful. This personalized approach would avoid the problem of immune rejection and could provide a source of perfectly matched rejuvenated cells for repairing almost any tissue in the body. Among the many exciting frontiers, one discovery stands out: cellular reprogramming. This is precisely what Japanese researcher Shinya Yamanaka achieved in a landmark study published in 2006. He found that introducing just 4 specific genetic factors into an adult mouse skin cell was enough to reprogram it. These cells were named induced pluripotent stem cells, or iPSCs. These 4 genetic factors, now famously known as the Yamanaka factors, act like a factory reset. The cell regains pluripotency, the remarkable ability to develop into any other cell type in the body, from a beating heart cell to a complex neuron. This discovery completely revolutionized biology. It proved that cellular identity is not a permanent state, but a reversible one. It was the biological equivalent of time travel. demonstrating that you could take an old, specialized cell and make it young and full of potential again. The initial method had a significant drawback. It completely erased the cell's identity. The key challenge, therefore, was to find a way to harness the rejuvenating effects without the dangerous side effects. This led to the development of a more nuanced approach known as partial or transient reprogramming. Instead of leaving the Yamanaka factors turned on continuously, what if you just pulsed them on for a very short period? A day or two? The first stunning proof of this concept came from the Salk Institute in 2016. The treated mice lived 30% longer. They showed signs of rejuvenation across multiple organs. Crucially, their cells did not lose their identity or form tumors. In my own lab at Harvard, we have shown that a modified cocktail of 3 of the 4 Yamanaka factors can restore lost vision in old mice. We did this by reactivating the cell's ability to regenerate their nerve connections to the brain, essentially resetting their epigenetic age and restoring a youthful function. In the immediate near term, over the next 5 years, the primary focus will be on safety and establishing robust biomarkers of aging. Simultaneously, the first in-human safety tests for partial reprogramming will likely begin on localized and easily accessible tissues, like the skin or the eye. The midterm horizon will be characterized by the approval of the first genuine health span extending drugs. In the realm of cellular reprogramming, research will advance from localized tissues to tackling entire organs. Therapies will likely become combinatorial. As we enter the long-term phase, the 2040s and the 2050s. The primary goal, safe and effective systemic reprogramming. An 80-year-old might have the biology, resilience, and appearance of a 30-year-old. We began with a dream and moved it into the laboratory. Current breakthroughs are providing an astonishingly powerful toolkit, but one tool stands out: cellular reprogramming. It suggests aging is often loss of information, cellular software corruption we can learn to fix. That makes the 2040s, 2050s timeline a plausible, if ambitious, bet. Beyond the technical hurdles lie a vast and complex landscape of social and ethical challenges. Who will have access? We risk creating a new, biological divide in society, a world of the long-lived and the short-lived. These are not questions for scientists alone. They require a global dialogue. The potential to alleviate the immense suffering caused by age-related diseases is a moral imperative. The science must move forward. However, this scientific pursuit must be paired with an equally vigorous and open conversation about its implications. We are arguably the first generation in human history to have a realistic chance of seeing the end of aging as we know it. This is an immense privilege and an even greater responsibility. The future we build should not just be a longer one, it must also be a better and fairer one for all.